Hello everyone, and welcome to today's Lab Tools webinar. I'm Nathan Nee, Associate Science Editor for The Scientist, and I'm here to introduce our discussion. Today, our speakers, Dr. Kit Fuhrman and Dr. Anushka Dikshit, will present on how to obtain whole transcriptome data from spatially mapped neuron populations. We'd like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and our speakers will address these questions during the live Q&A session following the presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. This webinar will be archived on the Scientist's website, and we will send you the link for the on-demand version via email within a couple of days. I'd like to take this opportunity to thank our webinar sponsors. ACD, a Bio-Techne brand, shortens the path to personalized medicine and enables research, drug development, and clinical applications by unlocking the power of RNA. ACD is a pioneer in the advancement of spatial genomics and a leader in the emerging field of molecular pathology, developing cell and tissue-based research and diagnostic tests for personalized medicine. NanoString is a leading provider of life science tools for translational research and diagnostics. Cited in over 4,000 peer-reviewed publications, the nCounter analysis system measures gene and protein expression to profile novel biomarkers. The company's GeoMX digital spatial profiler enables highly multiplexed spatial profiling of RNA and protein targets in a variety of sample types, including FFPE tissue sections. Please make sure to check out the downloadable materials provided by our sponsors located in our handout section on the right side of your screen. You can access and download these documents at any time during the webinar. With that, let me introduce today's first speaker. As the product manager for GeoMX RNA portfolio and discovery applications, Kit Fuhrman develops cutting-edge assays for the digital spatial profiler. He released the Cancer Transcriptome Atlas, the first high-plex NGS-enabled product by NanoString. Building on that success, Fuhrman launched the Whole Transcriptome Atlas for GeoMX. Fuhrman is currently building the next generation of spatial profiling tools and reagents for the scientific community. Prior to his tenure at NanoString, Fuhrman received his doctorate from the University of Florida in immunology, studying the role of regulatory T cells in type 1 diabetes, as well as his master's from the University of Central Florida, studying structure-directed design of HIV entry inhibitors. Let's just make sure everything is up and running. Dr. Fuhrman could just please share your screen before the presentation starts.
Good morning, good evening, good afternoon, everybody. Can you hear me all right?
Yep, you sound good, Kit. Can you just click hide on that bar and you're good to go?
That sounds great. Thank you, everybody, for attending today. I'm excited to present to you alongside ACD. Today, I'm going to talk about our GeoMX DSP platform and some of the assays that we have available to run on the GeoMX profiler. So, GeoMX is all about learning about the structural aspects of your tissue and profiling different regions with, high plex assays. And so here we're looking at a sagittal cross-section of a mouse brain. And this is actually an image from the digital spatial profiler. And so what it allows you to do is image a tissue section and then select different regions. And so here we've selected the hippocampus region, and it allows you to profile different regions of your tissue, in this case, the brain, in a myriad of different ways. And so you can do things like, unbiasedly grid a particular tissue section, or even better, you can actually select different structural features of the tissue of the brain, and then have individual protein or gene expression profiles for each of these. And so we've built reagents to run on this platform that allow you to get rich, high-plex information from various regions of interest. And so this technology has been on the market for about 3 to 4 years. In that time, we have, I think the number is now over 80 different peer-reviewed publications to date. So a lot of scientists have been using this technology to spatially profile their tissue and really get into the spatial biology work that they're doing in the tissues and learn a lot of great information. So a lot of great publications with our protein assays and our transcriptome RNA assays, and many of them using RNAscope as well. So to give you a sense of how it works, we provide the high-plex reagents, either protein reagents that have been labeled with the DNA barcode, DNA oligo, or RNA reagents, ISH probes that have the barcode indexing oligo on it. These reagents are used to stain your tissue along with morphology reagents, which we'll get into in a second. One of those choice options being ACD's RNAscope probes. The samples are run on the instrument, and you get what you see here on the top right is a really nice image of your tissue, able to visualize different regions and different structural features throughout your tissue section. And then the instrument will actually cleave the DNA oligos, and you'll be able to count and profile each individual region. So every circle on the right-hand side becomes its own unique protein or RNA expression profile. And so you're able to collect information at all of these different regions. The platform itself is compatible with a lot of different sample types. And so it really opens the door for a lot of different types of research and a lot of different types of archival tissue. So many people have FFPE tissue blocks that are in their archives, pathology archives, and you can use those on this platform, for both the protein and the RNA assays, as well as core needle biopsies, fresh frozen tissues that you may be using for other applications, things you've already slide mounted, and tissue microarrays, which are great for creating very large data sets across a lot of different patients. And so all of these sample types are able to be run on the DSP, Genomics DSP platform. And to give you a sense of the workflow, we start off with our high-plex reagents, either the protein reagents, like I said, or the RNA reagents that target the RNA transcripts. And each of them have this DSP barcode on them. The key point here is that it has a photocleavable linker between the probe and this DSP barcode, which is used in the region of interest selection. And so the user will start off by staining their tissue with these high-plex reagents and the morphology markers. So you can use antibodies with that to target different structural features, or you can use RNAscope probes to do that, or you can do both. We have applications where users have used both antibodies and RNAscope probes to target either RNA or protein to visualize the tissue. So once that's done, you've stained it with these morphology markers, you've stained it with one of our high-plex reagents. The sample is stained and run on the digital spatial profiler. And that presents you with a very beautiful image of your tissue sections, where you then select your regions of interest. And then once that's complete, the instrument will go to each different area that you've selected. It will shine a UV light to release those DSP barcodes. And then it immediately collects this DSP barcode into a zipper and deposits each of those into a well in a 96 well plate. So every single area that's been illuminated and cleaved gets put into a individual well in a 96 well collection plate. And then that's repeated for every single area that you've selected. So that process is repeated again and again for each area. And then it's read out on either a nCounter or a Illumina sequencer for our high-plex RNA and protein assays. So I want to dive a little bit more into the way in which we're selecting these regions of interest. We use a digital micromirror device to basically shape UV light to cleave the DSP barcodes from specific regions. So this allows us to be very precise in the compartments that we're selecting. And so you could do things like draw polygons and select circles and squares and irregular shapes of the regions you want. And so here I'm just showing an example where we've drawn a circle and we're getting out the whole transcriptome profile of that particular circle. But, you know, this is very informative. It can tell us a lot of things, but it is a mixture of tumor and immune cells. So with the DMD device, we can actually split it into two different profiles. So we can illuminate twice. We can illuminate the blue region, and then we can illuminate the yellow region. And we actually get two distinct profiles, one of those immune cells that are attacking the tumor, and another of the tumor, and see how they're responding to those immune cells. So a lot of rich information about the biology in any sort of tissue. So this is a great example of how you can use those morphology markers to segment on your regions of interest and really understand how the different structural features of the tissue that you're working with relate to the transcriptional protein function and expression levels. So there's a number of different profiling strategies that are available to our users. So geometric, as I said, segmentations where you're looking at large compartments and different cell lineages. Cell type, it takes it a lower deeper where you're looking at specific cells in a region of interest. So creating a nice bounding box and then selecting on, say, CD3 T cells or a particular neuronal cell type. Contouring, where you're looking at profiling between proximal and distal locations. And then a little more unbiased with a gridded approach. So all these are available when selecting regions of interest with your tissue. Now, just to show, we have a number of protein panels, validated protein panels that we've developed for our platform, and they cover immunology, oncology, and neuroscience. So a lot of different options, and users have the ability to mix and match our different modules across all three subject domains. And then just talking about total plex, you can actually mix these together with the NGS readout and actually read out over 100 different targets all at once. So a lot of great protein information. And just to give you an example of how contouring might work in, we have a user, Stefan Prokop at University of Florida or Pennsylvania this time. Basically, he was looking at myeloid cells expressing TREM2 in particular plaques in these AD patients. And so there are various differences between Alzheimer's dementia disease patients in the frontal cortex who have different TREM2 variations. And so the goal was to look to see how the brain and the immune system was responding to these, different plaques that are forming, and what are the differences between patients who have different TREM2 mutations and variations. So to give you a sense, this is what a plaque looks like in an Alzheimer's disease patient. And so we've imaged it with a number of different fluorescent markers. And then we've selected 4 different masks, as we call them, so regions of interest. And we have the center amyloid plaque, and then we create concentric rings around that plaque. So you're actually getting a contour of gene expression from close to the plaque to far away. So you can see how the plaque and the other neuronal tissue are interacting with one another. And so you can see you have key genes here of Alzheimer's disease being expressed in close proximity to the plaque, and those other genes that are expressed farther away from that particular plaque. And you get nice, I think here, protein expression or gene expression. And you can do that for all of your different patient populations. So you see, you can do a number of different plaques across the brain section that you're working with. So you can get, you can see how it varies across an individual patient. And you can also look across different patients with different TREM2 variations. We also have our high-plex RNA assays. So we've started off with the Cancer Transcriptome Atlas. It has all of our gene sets that we've developed on our nCounter bulk gene expression platform. So it covers quite an array of different immune oncology focused gene sets. Excuse me. And it is read out on an Illumina sequencer, and it allows you to customize and add up to 60 genes of interest. And this is just a sense of what's available. Some of these may be applicable to neuroscience. So we do have some users who will use it in certain contexts. But for neuroscience, many customers look towards our whole transcriptome atlas, which covers all of the transcripts in the human or mouse transcriptome. So all protein coding genes. So well over 18,000 for human and over 20,000 for the mouse genome, which actually has more protein coding genes. It can be used with fresh frozen or FFPE. So a lot of options for the user and a high level of sensitivity. So, typically over 9,000 genes detected per genes per segment or compartment that is selected. So there's a lot of great advantages. We're not biased towards the 3' end. So it's designed towards specific mRNA sequences. And so it's not dependent on the pulling down a poly A region where we have sensitivity to anywhere on the transcript and detect them. Very good sensitivity. Like I said, we can detect low, medium, or high expressing targets. You can use RNA scope or antibodies to define your regions of interest. And it works with various array of different sample types, FFP and fresh frozen, or fixed frozen if you're working with mouse tissue. And we have people who are adding in custom targets like synthetic transcripts that they're looking for in their particular tissues that they're working with. So, and it's read out, like I said before, on a Illumina sequencer and with minimal sequencing requirements. So one thing to look for when you're working with these type of assays is reproducibility, because you want to build, large data sets across tissues and patients and samples. So it's very reproducible from slide to slide, from user to user, and instrument to instrument. And so we've done a lot of validation studies to show that you can measure your biological signal with great confidence. You can also look across different tissues. We've worked with hundreds of different tissue types in human and in mouse. Here's an example of an FFP tissue array. We've actually repeated this for a number of different mouse strains. But you can see there on the right, for all those mouse strains, you can see the number of genes detected. It varies depending on the organ that you're working with, but some tissues have more transcripts expressed in the regions than others. But you see that diversity here on the right-hand side of the genes detected above the limit of quantification. So it gives you a lot of rich information of what's going on in each tissue. Now, to dig down a little more, we have a lot of options when running GeoMx DSP on neuronal tissue. These are two different mouse sections that we've run on the DSP. And on the left-hand side, we have the Allen Brain Atlas images. And so, the brain is really run by structure and function. And so each different structure actually correlates to a different functional unit. And so we can use the Brain Atlas to help us guide the region of interest selection on our mouse tissue, and then look at the transcriptional profiles and how they change from region to region. And so this is actually an earlier proof of principle experiment that we did in our R&D labs, where we simply just stained with DNA and then used the Brain Atlas to select the different regions. And so using the Brain Atlas, can we see differences in the biology in different structural features of the brain? This is actually with fixed frozen tissue, but it works just as well with FFP. We've done that separately. So we do see that we have differential gene expression between the corpus callosum and the Purkinje layers. All of the different regions that we select. We can really see the different layers using the density of that DNA stain. And then we can basically find differential gene expression, and key markers for every region is expressed at the right places. We can also see that the Brain Atlas allows us to look at the protein expression, and we can see how in particular regions how proteins are expressed, and then recapitulate that using our profiler. And then we can see how the different transcripts that correlate to those particular proteins are expressed in the regions that we've drawn here on the right-hand layer in the cerebellum. And so we see that, structure follows function, and the genes that, are expressed at the protein level are also expressed at the transcript level in individual structural regions of the mouse brain. And so I just want to kind of end my portion of the talk talking about our relationship with ACD. And Anushka will kind of go into more detail in the field of neuroscience, but we've developed a lot of different kits. This is an example of some probe combinations that we've worked with in oncology, but we've worked on several different combinations in neuroscience as well. You can use RNAscope to visualize up to 3 targets plus the DNA stain with DSP. You can use these predefined kits that we kind of worked out, whether in neuroscience or oncology, immuno-oncology, or choose your own targets. So you're not limited to what's here. We've really validated the whole protocol with DSP. It can be automated, and it can work with our GeoMx RNA assay, so the Cancer Transcriptome Atlas and the whole transcriptome assay. So what it allows you to do is select your regions of interest based on the transcript expression levels that you're looking at. So, many times there are particular markers for different cell types that are best visualized with an RNAscope probe. Maybe the antibodies that aren't good, or maybe it's just a simpler system if you're already working with particular RNAscope probes that you want to use in your project. And you can build regions of interest and look at differential expression between your different regions across your tissue. And here, this is just an example of a small cell lung carcinoma and differential expression between 2 different regions. So with that, I'd like thank you, and I'll pass it off to Anushka.
Thank you very much for that presentation. As a reminder to our audience, please do not forget to submit any questions that you might have for our speakers using the Q&A tab at any time. And as Dr. Fuhrman said, our next speaker is Dr. Anushka Dikshit, who is the Senior Application Scientist with Advanced Cell Diagnostics, a Bio-Techne brand. She has a background in cancer research and immuno-oncology, and is focused on expanding the applications of RNAscope technology for oncology research. Prior to joining ACD, she was involved in identifying novel therapeutic targets for melanoma as a postdoctoral fellow at Duke University, and she obtained her doctoral degree from Southern Illinois University School of Medicine in molecular physiology. Let's just double check if everything's up and running. Yep, everything looks like it's good to go. Dr. Dikshit, thank you very much. The floor is yours.
Thank you so much for that introduction. Can you all see my screen okay?
Yep, it looks great.
Excellent. So, as Kit mentioned, RNAscope in combination with GeoMx can really help expand the applications of spatial analysis with that tissue context. For today's presentation, I'll be giving you a background about the RNAscope technology and then move on to show you a couple of examples of how this research has been applied, how this technology has been applied in neuroscience research. And following that, I'll be also talking about an example of how RNAscope GeoMx DSP joint workflow has enabled looking at specific subpopulation of neuronal cells in different regions of the brain. So to start with, for those of you who are not aware of this technology, the RNAscope assay is an in situ hybridization platform with specific probe design and amplification strategy that allows for visualization of your target RNA marker with high level of sensitivity and specificity. So what makes this assay unique is that this is an ideal spatial analysis solution to interrogate complex tissues. It is very specific and sensitive, and therefore can be made applicable to a range of sample types, which include FFPE tissues, fixed and fresh frozen tissues, PBMCs, and cultured cells. Additionally, our technology has been developed such that we can design very specific target probes against a range of RNA targets. These include mRNAs, long non-coding RNAs, microRNAs, ASOs, splice variants, highly homologous sequences, and so forth. And as you can see here, our downstream signal detection strategy is also extremely robust, where we can actually visualize a single molecule of RNA at that single cell resolution. So currently, we have 4 major assays that enable you to look at the target of your interest based on what your research question is, starting with the RNAscope assay, which is our parent assay designed for targets that are over 300 nucleotides in length. Then you have the BaseScope assay, which is more of a specialized assay for targets between 50 to 300 nucleotides. And then we have our microRNAscope assay, which is for even smaller RNA molecules, like your microRNAs, siRNAs, and ASOs, which allow detection of targets between 17 to 50 nucleotides in length. And finally, we have our recently developed and launched DNAscope assay, which transfers our technology onto detecting a different biomolecule, which is the DNA now, where we can look at target genes within the genomic DNA. This assay was mainly launched to expand the applications into looking at gene fusions, translocations, amplifications, and deletions. So overall, depending on your research question, you can utilize any one of these platforms to help advance your research. In the next slide, I'm just going to go over some of the strategic elements of our technology that make it stand apart from some of the other tools. And we take extreme pride in our probe design strategy. As you can see on the screen, the probe we depict as a Z molecule, which identifies different parts of the probe, namely the target binding site, which is the part of the probe that binds onto your target sequence. So it's complementary to your RNA target. Then we have the preamplifier binding site, which forms the basis of developing the amplification tree. And then we have the linker region, which combines or joins these two regions. So the way we design our probes is that such that a pair of Z probes have to bind together in tandem onto the target sequence. And we design probes as a set of pooled probes. So you'll have these ZZ pairs bind along the target sequence. Once the Z pair is bound, we have the preamplifier molecule that comes and binds on to the pair of the Z. Then you have your amplifier, and then you have your labeled probes. Now, these can either be chromogenic or fluorescent based on the assay of interest. Now, this amplification strategy ensures high level of sensitivity, allowing detection of even the most low expressing targets. Then we also ensure that our probes are highly specific, because unless you have a pair of Z's bound tandem to each other, you won't have the preamplifier binding on to the Z probes. So if, for example, you have a single Z probe non-specifically binding onto a sequence, then the preamplifier will not bind to the Z probe and there won't be any amplification tree formed. This ensures that high specificity of detection, giving high signal to noise ratio. So overall, we have expanded our portfolio to incorporate different plex levels based on your research question. You can perform single plex or up to 12 plex for FFPE tissues and 48 plex for fixed and frozen tissues. Ultimately, because of the flexibility that we offer in our sample type and applications, we have been used for a wide range of applications and in a range of research areas. Some of the key applications enabled by this technology include detection of non-coding RNAs, where using an antibody obviously is not an option. Looking at splice variants, so when you want to identify multiple isoforms of a gene. Looking at CDR3 sequences on T cell receptors to identify clonal populations of cells, and so forth. One of the major applications which I'll be going into a bit of detail today is validation of single cell sequencing. But overall, we have been utilized in basic science research, infectious disease research, cancer and clinical research, and biopharma discovery research. As I mentioned, the focus of today's presentation is going to be on neuroscience research, and I'm going to be focusing on a couple of applications within that research area. Overall, our technology has been widely applied to look at targets such as GPCRs and ion channels in the brain where you don't have a good specific antibody to use. Secondly, we have been extensively used to characterize different neuronal cell types by visualizing co-expression of targets and studying their activation states. Now, this becomes extremely important in pathological samples when you're trying to determine a molecular mechanism underlying a certain disease. Then ultimately, because we are an RNA detection technology, you can visualize secreted factors and look at plasticity markers and identify the cellular source of these secreted molecules. And finally, as I said, we have one of the biggest applications for spatially mapping single-cell sequencing data downstream. We have been considered as the gold standard, and a lot of these highly multiplex technologies are using our platform as a downstream validation tool. So today, I'm going to show you an example of how our HiPlex assay has been used for validation of single-cell sequencing downstream. So here's a standard workflow of how you would go about performing single-cell sequencing to profile a tissue or identify specific neuronal subpopulations within the brain. So your blood tissue will be dissociated into single cell. You will have that single cell gene expression information, and then you'll identify certain key targets that you want to visualize with a spatial context, and then perform the HiPlex assay. Now, this forms a nice orthogonal validation tool to visualize any novel cell types that have been identified by single cell sequencing, and also visualize the target genes expressed within these cells. So I'm going to show you an example of how we validated a paper that came out in 2016 that looked at the median spiny neurons in the mouse cortex. And you have two major subpopulations within these neurons, the D1 MSN neurons and the D2 MSN neurons. Each of these subpopulations have a specific gene signature, which is marked by these targets showed on the screen. And within these subpopulations, you have subpopulations of these neurons, which express specific combination of these gene signatures. And so we found that using the HiPlex assay is a perfect solution to visualize these subpopulations and their subpopulations within a specific region of interest within the brain, mouse brain cortex. So we designed probes and combined these 12 targets, 8 of which were specific for the D1 and the remainder specific for D2. And then we took 5 and 5 of each of them and combined them with 2 targets that are commonly expressed in both D1 and D2. And here is the result from that, where you can very specifically study where each of these targets are expressing within that region of the brain and identify how the D1 and D2 populations are organized within the mouse brain cortex. So this allowed us to nicely profile these neuronal subtypes within the mouse brain cortex. And with our registration software, you can actually visualize all of them together and toggle between these genes to only study the specific subtype of your interest and understand how they are spatially oriented within the brain. So after this example, I want to show you another unique example using our microRNA scope assay about how you can identify key microRNAs that are important in regulating certain neuronal functions and important for developing embryos within the mouse brain. So I'm going to show you a few examples of how our new microRNA scope assay was able to detect these target microRNAs within these mouse brain tissues. So here's an example where we have shown expression of target microRNAs like miR124, LED7B, and so forth in the mouse brain. And the key thing to highlight here is that you can pick up these specific microRNAs that only express in those target cells within the brain at a certain stage in the developing phase of the brain. And here we can pick up miR124, which is one of the most ubiquitously expressed microRNA in the brain, LED7, and then miR138 specifically expressed in the target regions. And then we have Scramble control just showing the specificity of these probes in detecting the target microRNA of interest. Furthermore, we also wanted to show that this assay works really well across species. And here we are focusing on just miR124, which is known to express across different species and specifically in the brain of different species. And here we are comparing mouse brain, rat brain, and human brain. And you can see some nice robust expression of miR124. But when you look at mouse liver, which we do not expect miR124 to express, you can see negative signal for this particular marker, indicating against how specific we can be with respect to these probes. So this is really important when you're trying to identify targets or trying to understand an underlying mechanism of certain pathological conditions. And in this next figure, I just want to highlight again the specificity of this assay, where we are looking at miR124 in this developing mouse embryo. And you can see how nicely it's expressed specifically in the brain region, and then some part in the developing spinal cord and so forth. But this really emphasizes on how important spatial analysis can be, because it helps you not only visualize your target, but understand the functional importance of that cell type or organ system when you're studying certain pathological conditions. So with that, I just want to highlight how important this technology has been and how well it has been adopted over the years. We have over 5,000 papers currently in peer-reviewed journals, in top-tier journals across different research areas. Some of our key research areas being neuroscience and oncology research. So in the next part of my talk, I want to focus more on how our two technologies, the GeoMx DSP technology that you heard about from Kit and the RNAscope technology that I just introduced, can be combined to provide a highly multiplex spatial solution that enables studying complex tissues like the brain and the tumor. And just show here an example of how we profiled a mouse brain for studying different neuronal subpopulations in functionally relevant regions of the brain. So with that, here I'm showing you a standard workflow of how the RNAscope assay can be combined with GeoMx DSP. So the RNAscope assay falls upstream of the GeoMx DSP and acts as a molecular guide to identify regions of interest that can be further profiled by GeoMx DSP. So shown here, we wanted to see how well these probes work with the GeoMx DSP. So we started with picking target probes for V-GLUT and VGAT markers, which are your markers for glutaminergic and GABAergic neurons, and see a general spatial distribution of these neurons in the sagittal section of the brain. And you can really appreciate how well this technology works in giving you that spatial context of presence of these target neurons across the brain. So with this particular assay, you can then hone in on specific regions of the brain to further profile using the GeoMx TSP, which then provides you that transcriptomic information for that particular region, where you can study thousands of targets at the same time. So for our study, what we did here is we actually utilized the RNA-scope protein co-detection assay upstream of the GeoMx. And the goal, again, was to look at the different neuronal and non-neuronal cells within the region, within different regions of the brain, and then profile these regions based on the expression. So we started by looking at a couple of RNA and protein targets. We looked at the V-glut RNA target in combination with the Neuron antibody. Similarly, we looked at the V-glut1, V-glut2 RNA-scope probes along with the TMEM119 antibody, which is a marker for glial cells, as you know. So just to highlight the protocol for this co-detection assay, you first add your antibody and then add your probes, which after hybridization, you develop the signal for both of them and then visualize them simultaneously. So this workflow was done upstream of GeoMx. And what we found was that there was very strong correlation that we observed with the co-detection assay and with the standard GeoMx. Just making sure that these chemistries are not interfering with each other and the workflow for the co-detection is compatible with the GeoMx assay. So after establishing that, we decided to go with the combination of the V-glut1, 2, and VGAT probes with the TMEM199 antibody upstream of GeoMx. Now, what this helped us do is it helped us profile microglia in combination with the neuronal populations. And depending on the distribution of the cells within each of the ROIs, we were able to profile 4 different cell populations within a given region. So as you can see here, based on the expression pattern, we either saw excitatory neurons or inhibitory neurons or microglia. So we were able to identify each of these populations within a given region of interest and then profile this region for transcriptomic analysis using the GeoMx. And what we found was that we were able to identify up to 3,000 targets on an average for the microglia population and over 8,000 targets that were expressed within the neuronal population of the cells. So the next step was to confirm the specificity of the cell types that we had identified using this joint workflow. And so to do that, we did spatial cell type deconvolution using single cell sequencing data that is available from the Allen Bain Institute. And they have information about the specific cell types. And what we did was we compared that database information to the transcriptomic data obtained from the GeoMx analysis. And as you can see, microglia-specific markers are expressed by the microglial population that we identified by the GeoMx TSP analysis. Similarly, cells specific to the neurons, again, were found in the neuronal populations that we profiled using the joint workflow, just showcasing that we were very specific and sensitive in identifying these different population of neurons and glial cells within these specific regions of the brain. And finally, we wanted to identify differences in gene expression in the neuronal populations within an array of functional regions within the brain. So how these same population of neurons have differential gene expressions based on which region of the brain they're present in. Because each of the neurons present in amygdala versus the hippocampus or the cortex have specific function to serve and have specific transcriptomic profiles. So here you can see when we're looking at correlation of this target with the different excitatory and inhibitory neuronal targets, we find that the correlation is different based on the different functional region of the brain. And that was true for a couple of other targets that we identified. And here we are looking at some targets that are involved in and are known to be differentially expressed in the autism spectrum disorder and in schizophrenia. And so going further, we again showing here these targets, how they're differentially expressed in different regions of the brain. We are focused here on the amygdala, and we're demonstrating how, for example, NEO1 and RELN are expressed in, differentially expressed in these different regions of the brain. So overall, what these images are showing is that specific regions of interest and cell populations show elevated expression of these three key targets that we're highlighting here. And any future experiments we do on profiling these cell populations will be critical to understand, cell-specific information based on their spatial location within the brain. So overall, this study, I think, gave us a really good idea about how well the joint workflow is in studying targeted expression within specific regions of the brain, and then tying them to the functional significance of that brain region. So in this example, hopefully, we've shown you that geomics WTA analysis can help profile up to 17,000 targets in mouse brain. Using the RNA-scope protein co-detection assay, we were able to look at RNA and protein targets, which acted as molecular guides to then select target regions of the brain that were further profiled by geomics DSP. And so by using pathological models, we can actually study, targeted, we can study targets that can eventually be used for therapeutic purposes and provide, functional information in certain regions of the brain that are still undiscovered and novel. So there are a lot of applications of this joint workflow in neuroscience and in many other research areas. So with that, I want to thank you for your attention, and we'll take any questions. Thank you.
Thank you very much for that presentation. Before we start our Q&A session, I just want to give a final reminder to our audience that to submit your questions, please use the Q&A tab. And we do have several questions in the queue, so let's get to them. Our first question will be for Kit, and that question is, what's the smallest size of the ROIs you can use? And I suppose as a follow-up, we can ask Anushka too, how does the size of ROIs impact compatibility with single-cell analysis?
That's a really good question. So the DMD mirror device on the digital spatial profiler, the Geomics DSP, is highly tunable down to 10 microns, and can be used to illuminate a single cell. But it's it ends up being all about how much you are actually going to be collecting from your particular regions of interest. That translates directly into the amount of material that you're going to read out. And so, the typical applications with DSP are looking at, the tissue level, right? Looking at different structural features across the tissue. And typically people will look at kind of functional units like islets in a pancreas or neuronal regions in the brain, different specific structural regions in the brain. With that said, there are some people who will image larger cells and that have enough mass to be detected. The way to take your single cell analysis and translate it to geomics is using the single cell gene signatures that you've developed in your single cell analysis. And then you can use that to basically deconvolute the cell populations that are in any particular regions. And so we've published a number of different gene signature sets online on GitHub, but you can also use our algorithms and take your analyses, your single cell analyses, and use that to define the different populations so that it can be applied to any sort of functional study that's going on, any tissue or regions or cell types you want to look at.
All right, thank you very much. For our next question, Kit, going back to Kit again, For DSP, for detecting protein levels, do the oligoconjugated antibodies have to be commercialized?
Yep.
Or can you use custom antibodies from somewhere else, or maybe homebrewed or something like that?
Yeah, absolutely. Yes, absolutely. So we've done a lot of work to validate the antibodies that we have in our modules and in the different panels, so that you have confidence that when you use them in the assay, that they're detecting the proteins that you're expecting. There are many cases where you want to add in your own proteins of interest, and you can definitely do that. And definitely reach out to us if you want to use your own antibodies. There's a number of different routes to go about doing it, but yeah, yes, you can definitely add your own antibodies into the assay.
Thank you very much. For a question for Anushka, could you introduce the difference between RNAscope and HiPlex in their working principle, and why can HiPlex detect more genes simultaneously than RNAscope?
Thank you. Yeah, that's a great question. So in principle, the HiPlex is part of the RNAscope assay technology. So it is, the name of the assay is RNAscope HiPlex. So with this assay, what we've done is we've expanded the capability of how many targets you can detect simultaneously. Principally the assay is exactly the same. Chemistry is pretty similar. The only thing is, with this assay, we have introduced the iterative modality of detection, where it's cyclic detection. So you do 3 rounds of staining so that you have all the 12 targets on the same section of the tissue. So that is the main difference. The probe design strategy and everything remains the same. The sensitivity, specificity is very much similar to our original assay.
Thank you very much. Next question for Kit. What is the status of cellular deconvolution for geomics data in neuroscience applications?
Great question. So we do have some basic cell type signatures that are available, which if it's not already on GitHub, it should be going on GitHub rather soon. So we have the basic populations, and of course, like I said before, if you have specific populations of neurons or microglia that you're actually working with that you want to define yourself, you can use those, or you can use our preset ones. So if it's not already on GitHub, it should be on GitHub very soon. You can look at, you can find it through our website through going to our geomics tools page and navigating to the GitHub repositories.
Thank you very much. Next question for Anushka. Can the co-detection assay be used on an automated platform?
Yes, the one that I demonstrated here in the presentation was also done on a automated platform. So we have this assay available in multiple configurations. The one I showed today was with our multiplex assay, but you can do it with any of our chromogenic assays as well. And it's available in both manual and automated formats.
Thank you very much. Next question for Kit. What is the maximum number of colors that can be run simultaneously? Alternatively, what's the maximum number of masks or segmentations that can be introduced simultaneously?
That's a good question. So when selecting your regions of interest, the physical instrument itself has 4 fluorescent color channels. And so, you can use up to 4 different fluorescently labeled morphology markers, detect your regions of interest. Taking that one step further, if you have, other images on serial sections or things you've done on other microscopes or on other scans, you can actually overlay those images on top of the image that you're working with on the DSP. So 4 layers immediately on the instrument, but then you can add more information to selecting your regions of interest using the overlay function. Kind of moving towards the ROI and masking strategies, you can do as many combinations as the number of fluorescent channels will allow. So combinatorially, that can come out to quite a bit. I think in practice, most people do maybe about 4 to max, maybe 5 in rare instances, but different mask within a single region of interest. So there's quite a bit of flexibility there. Practically, you're probably only going to do, probably 2 to 5 different masks, depending on how many morphology markers you're using at once.
All right, thank you very much. Another question for Anushka. Can the high-plex assay be used on FFPE tissues?
Yes. So when we originally launched our assay, that was for fixed frozen tissues a couple of years back. But we recently launched a newer version of the assay, which is now basically the same assay with expanded applications on FFPE tissues as well. So the same assay can be used for fixed, fresh frozen, and FFPE tissues. And we've really taken care to ensure that autofluorescence is not an interfering factor with this assay, which is often the case with a lot of FFPE samples. So we've really optimized and tweaked the technology to ensure that we are minimizing the autofluorescence and you can see the real signal without any masking.
Thank you very much. A question for both of you, because you, and this will vary, I think, depending on circumstances and parameters, but how long does it take for the GeoMx to process tissues? So the question is actually, how long does it take for GeoMx to go, essentially to go through a 1 centimeter by 1 centimeter tissue section? Obviously, this will vary depending on parameters and what's being measured, but in a general idea, is it quick? Is it slow? That sort of thing.
Yeah, I think I can maybe take this. After you do the staining with the RNA scope probes and the GeoMx RNA assays, you'll be presented with the image of the whole slide. And so that scanning, the setup and scanning takes just about an hour. And so you have that image. And then you can select the regions of interest. I think to select, look at a centimeter by a centimeter, select your regions of interest. The collections only take, I would just give you a rough estimate of maybe an hour, depending on how many regions you select in that square centimeter. And then you take that collection plate and you will run it on the readout. So that may take another day or two, depending on how close you have access to the readout instrument, such as the sequencer or the nCounter instrument. So the time that it takes to collect, relatively short. I think the whole end-to-end assay can easily be done in a few days.
All right, thank you very much.
Good.
I believe we have time for one more question, and this will be for Kit. For GeoMx in the brain, is it possible to register brain sections to the atlas, and also to use atlas-defined regions to segment for transcript collection?
Absolutely. So you can take those images that the Brain Atlas will, that they produce, that kind of maps out the different structural regions, and then use the overlay function to basically figure out which regions correlate to which ones in your tissue segment. Depending on the scale of tissue you're working with, you might take different maps and overlay them. I think with the mouse brain, of course, you can fit the whole thing on a slide. In a particular cross section, human brain, you may have to narrow it into particular regions. I think maybe we're also working on applications where you can do a little more digital pathology applications, and feel free to reach out if you want more information on something a little more programmatic. But most of our users are using the overlay function to do that sort of thing.
All right, thank you very much.
Sure.
Unfortunately, that is all the time that we have for today. If you have any further questions, please consider reaching out to our speakers directly. Their emails are shown on the screen. As a reminder, this webinar will be archived on the scientist's website, and you will receive an email notifying you when the on-demand version becomes available for viewing. I'd like to thank everyone who took the time to join us today, and particularly those who shared your questions and your comments. On behalf of the scientist, I'd like to thank our speakers, Drs. Kit Fuhrman and Anushka Dikshit, as well as our webinar sponsors, ACD and NanoString. Thank you, everyone, and have a pleasant day.